Hi, everyone. I think this is Jishen Zheng from Westlake University. So I'm very glad to be the moderator for this section of methods of collection, preservation, and research. So in this section, we have four speakers. So each speaker have 15 minutes to present their work, and then we'll have 15 to 20 minutes to discuss and for Q&A. So first, I would like to introduce the first speaker, Dr. Niranjan Nagarajan from Genome Institute of Singapore. So his talk title is Strategy for Generating a High-Quality Gut Microbial Reference Database for Southeast Asia. So welcome, Niranjan.
Thank you.
You can share your screen.
Yes, I will do that. Can you see my screen now?
Yes.
Okay, great. Okay, so thank you for the kind introduction and good morning, everyone. It's really exciting to have this opportunity to present in this exciting symposium. My name is Niranjan Nagarajan. I'm from the Genome Institute of Singapore. And my talk today, as has been mentioned, is going to be about cost-effective ways to generate high-quality metagenomic references for Southeast Asia, drawing on some of the learnings that we have from a previous study. And the motivation really for doing this is the fact that microbiome studies are increasingly becoming a part of population health studies. So it's not just a small number of samples that we're trying to study, but we're trying to study the entire population. And a great example of this is the Lifeline study in the Netherlands, shown here with a large data set of more than 8,000 metagenomes, and this very nice recent publication that combined multiple data sources, environmental, patient lifestyles, and so on, to answer a key question in the field, which is how do we define a healthy gut microbiome? And then from that, perhaps understand its role in diseases. Another really exciting study in the space of population health studies is a component of the All of Us program in the United States. And here the motivation is to really understand how diets impact human health for precision nutrition, with the microbiome being an important modulator of its effects. Now, in addition to understanding of how the human microbiome impacts various disease conditions, this is a growing field. And so a prime example of this is really C. diff colitis, which is a classic example where microbiome therapy is already in practice. But there's also a fair bit that's known in terms of a few other areas, including immunotherapy and GI diseases such as IBD, where large-scale clinical trials are underway for various treatments, and as well as using microbiome as a biomarker for response. And finally, there are some several promising areas where we continue to accumulate evidence, including cardiovascular disease, skin, oral health, and the gut-brain axis. And some of this work actually comes from Singapore, showing that it's relevant in our part of the world. Now, the classical approach to study the microbiome, specifically the human gut microbiome, has been to use marker gene sequencing, but it has its known limitations, including the low resolution, as well as various amplification biases, depending on the choice of primers. And more recently, microbiome studies have adopted a shotgun metagenomics approach to address these limitations. And there are several advantages. Of course, the higher resolution, the species and strain level resolution, the information about pathways and gene function, the reproducibility, the ability to look at variations in the genome and evolution. All of these are strong advantages. But a lot of shotgun metagenomic analysis, especially for the rarer species, relies on genomic databases for profiling analysis. And the incompleteness of these databases is an issue. As we know, microbial diversity is huge, as shown in the tree of life. You have the bacteria on the top, and essentially all other forms of life, right? Archaea and eukaryotes are in the tail out here. And as you can see, all these clades with dots in this figure in front of them, they lack an isolate reference. And so when we start to use reference databases, we need to worry about the quality of the reference databases. And so, of course, one way you can try to address this is directly assemble genomes from metagenomic data. And a great example of this among many nice publications in this space is this one from the Sigata lab. But these studies rely on, and most of these studies have relied on short read assembly. And so this gives a genome in, sorry, I'm trying to have in hundreds of 10 kilobase pair or so fragments, and clustering these into a bin to get a genome can be error prone. So the fragmented nature of short read assembly can be a challenge when trying to study genomes obtained from metagenomic assembly. Also, these studies often require a large number of samples to get good coverage of various species. And finally, they have a sort of Western bias, because most of the data that comes for these studies, it comes from the US and Europe. So to address this question, and particularly in our neighborhood, in this part of the world, we started with what we call the Singapore Platinum Metagenomes Project. And this is a similar idea as previous Platinum Genomes projects, where the emphasis is on quality of the genomes that are produced rather than the quantity. And to enable this, often we try to combine the strengths of multiple technologies, including deep Illumina sequencing for base accuracy, long reads with the Oxford Nanopore platform in this case for its ease of access and ease of use, as well as high-C sequencing for a subset of samples for long-range connectivity information. And really, despite using multiple technologies, these are relatively easily accessible technologies, the first two especially, and the total cost can be perhaps just a factor of 2 off from the initial cost of just shallow Illumina sequencing. And we applied this approach to a modest size cohort. It's a multi-ethnic cohort that was already there in Singapore with Singaporean Chinese, Malay, and Indian subjects. And we tried to capture as much of the ethnic diversity in Singapore as possible here. Now, one of the questions we had to address was really the quality of long-read data, or the ability to generate long-read data for complex samples and complex matrices such as stool samples. And for this, we collaborated with Professor Hiening Seedorf at TLL, and he used this very nice sample collection approach that allowed us to preserve the viability of cells better, so to preserve them for further culturing studies. But also we noticed that this approach was really nice in order to preserve long read lengths. So we had N50s of nearly 10 kilobase pairs. So the sort of the median statistics of read lengths were close to 10 kilobase pairs. And we got several megabase pairs of data per sample, even though we were multiplexing sometimes 12-plex per MinI and flow cell. And the data also maintained a profile that we expected, at least was concordant with the Illumina data. So if you look at the correlation between the Illumina and Nanopore profiles, we got very high correlation, 0.9 or around there. And this allowed us to assemble them together. The next challenge is really assembly. And in particular, we were worried or concerned about having hybrid genomes where there are multiple strains for a certain species, and we wanted strain-resolved assemblies. And for this, what was really useful was this hybrid approach where the short reads give you accurate assembly graphs that can then serve as a basis for long reads to disambiguate paths through the graph in the assembly problem. So instead of just using genomic coverage, which is a common approach to cluster context and get bins, we can directly exploit the assembly graph information shown here to split the metagenome into genomes and in a sort of segmentation problem. And if you're interested, I would encourage you to, into the bioinformatics, encourage you to read this paper that was published a couple of years ago. So this hybrid assembly, of course, because it uses long reads, fundamentally has an advantage in terms of contiguity. And it is a significant advantage when you think about studying these genomes, because instead of having tens of KB in terms of fragment size, we get hundreds, if not megabase size, hundreds of KB, if not megabase size fragments. And so this allows more careful analysis. But in addition to that, we saw that we recovered many more genomes. And this is a function of also of the binning approach, being able to recover high quality bins. We recovered many more genomes with this approach, even for the most abundant species in the sample. So some of the bifidobacteria or the ruminococcus species, these top 10 genera in the gut, they were much better recovered, as shown in blue here, compared to just the short read assembly with green. So we add hundreds of genomes per sample, hundreds of genome overall in this analysis. So when you use these high quality genomes as references, you immediately get a boost in your analysis for reference-based analysis. So in several samples where we use short read references, you will not detect the various bifidobacteria that are present in the sample, but these could be detected in the long read assemblies. So you can see that the blue dots for the long read based analysis, they are on as expected, while the green dots often you don't detect them, so they show abundance of 0. In addition, despite having much fewer genomes in our database, we call this the SPMP, the Singapore Platinum Metagenomes Project, which has a few thousand genomes in it, compared to UHGG and other public databases, which have tens of thousands of genomes in them. We find that the number of reads you map to our local population-specific databases is a boost, a 10% or so boost. And this, of course, is useful for various kinds of mapping-based analysis. Now, what was surprising to this for us was that when we then started looking in terms of the novelty that we had in our data, and despite Singapore being in a very urban, very westernized population, nearly 10% of the species level clusters that we detected from the 4,000 odd genomes that we had were completely novel compared to existing databases. And when we look at the level of strains, this is even more stark. Most of the strains, say the 99% identity threshold, are unique to Singapore. They are not seen in any public database, even among highly studied gut bacterial species like Bacteroides uniformis or Faecalibacterium proxenici. These are all very unique strains. And in particular, we were very curious to see the bifidobacteria diversity, because as you know, bifidobacteria are particularly important gut bacteria, that we described in the previous talk as well, in terms of functions, early colonizer, marker of healthy microbiome. They also have commercial value. So we were surprised that when you take the genomes that we have and compare them those two publicly available genomes, both cultured as well as uncultured, you find essentially entirely novel lineages that are only seen in our data, so as marked by these green triangles here. And you can see that many of the isolates, the green dots in these figures for bifidobacterium, they cluster in a few regions of the tree, suggesting that all the isolation work we have been doing is biased to some extent. And there might be important lineages and functions that we have really not explored in terms of bifidobacteria capabilities. Now, this was possible to some extent in our study because, especially for bifidobacteria, they're particularly enriched in Singaporean populations, not as much as in the Mongolian population, but they are enriched compared to, say, Western populations, and they seem to also have an ethnicity bias. So ethnic Indians and Malay have more bifidobacteria than ethnic Chinese. And so it's precisely because of this, we think it's important to study ethnically diverse populations to uncover interesting microbial genetic diversity. Now, at the functional level, we focused primarily on biosynthetic gene clusters because of the many functions and applications as antimicrobial peptides and other functions. And in total, we have 27,000 novel clusters among the thousands of others that we discovered, basically serving as a treasure trove that it can be enabled by high contiguity and quality of assemblies. And one of the annotations we did was to look at antimicrobial peptides and to fold their structures using AlphaFold. And we found some very interesting structures, elasopeptides and lanthipeptides, which had expected structures, but also some unique structures. So there's one example, we had 2 bacteriocins that were encoded in the same biosynthetic gene cluster, and they seem to form a sensible looking complex structure based on this AlphaFold predictions, as well as a very interesting lactococin here, the beta sheet structure and this long tail. In particular, we found a very novel bacteriocin in a common Bloschia species. So this Bloschia species is essentially present in most of our subjects, but it has different organizations, but they all retain the export protein and the peptide modifier. So this seems to be a functional biosynthetic gene cluster. And when we looked at gut microbiomes that contain these strains and with this BGC and without this BGC, we get very different correlation structures. So the correlation in the abundance of the various bacteria is very different. In one structure on the left with the bacteriocin, we see that the Bloschia species seems to inhibit various Faecalibacterium processi types, but without it, it doesn't. And this, we think, can fundamentally change the structure of the gut microbiome. So this seems to provide us an example of where a bacteriocin might be playing an important role in shaping the microbiome. And moving forward, we are also interested in looking at other things like phage diversity as a way to modulate the microbiome. We think the high quality of the assembly really allows us to identify a large number of putative phages and use the CRISPR sequences in the host to associate the phage with their host. So we're studying this further now. We watch the space for new results. With that, I'd like to thank you for your attention. I will leave you with a few takeaways. Hybrid assemblies are useful for high quality assemblies from metagenomes. And there's a lot of value in developing country-specific, in fact, population-specific databases. It's particularly important for large and diverse countries such as Indonesia. Finally, we cannot afford to ignore the dark matter, the unreferenced genomes out there. In fact, we are likely to uncover a lot of interesting biology precisely in this space. So with that, I want to acknowledge Professor Peter Little for really seeding this project and believing that there are unique strains in our populations, as well as my collaborators, Professor Y.Y. Teo and Professor Henning Seedorf, Jean-Sebastian, Ming Hao, and Amanda really drove this project. I'm really grateful to have them as part of the lab. And with that, I'll be happy to answer questions. Thank you.
Thank you, Niranjan, for the very, I think, high quality work, which is really inspiring. So I think we will leave the Q&A to the end of this section.